So in part 2 of chapter 19, we're going to be moving downstream from the glomerulus down into the proximal tubule, loop of Henle, distal tubule, and collecting duct, looking at reabsorption, that is the removal of material out of the tubule back into the bloodstream, and secretion, which is the deliberate inserting of material from the bloodstream into the urinary filtrate. One of those mechanisms is secondary active transport. We've talked about that a lot this semester. We're even going to show you some tertiary active transport in part 2B of this chapter. So let's get started. So a bit of a summary slide about what the nephron is doing in terms of filtration and so forth, summarizing a lot of what we've talked about up to this point and then showing you where we're headed. First of all, we have the glomerulus and we have this afferent and efferent arterioles, and I've put these little valves on here to represent the smooth muscle that we find on each of these arterioles. Now, these arterioles collectively will influence the hydrostatic pressure. Certainly the pressure coming from cardiac output and the mean arterial pressure is going to contribute, but these guys will modulate that pressure. So I think that's the hydrostatic pressure. We also have the plasma oncotic pressure, which is pulling fluid back into the capillaries, primarily through those albumin proteins that we talked about feels like a while ago, but yet still part of this unit. The third pressure that we deal with is called the fluid pressure, the pressure we find in Bowman's capsule and downstream. This fluid pressure is also opposing hydrostatic pressure. Usually doesn't amount to too much, but if we have any type of occlusion or injury or something along those lines, if fluid pressure begins to rise, it becomes a very, very serious clinical parameter. So that's why we talk about it here. But the sum of these three pressures should mean a net filtration pressure. Now, we have the macula densa as part of the juxtaglomerular apparatus here. The macula densa are these red cells right here. They sense flow and osmolarity in that order. If they sense a change in flow, they will modulate the constriction and dilation of these valves, and they work in opposite directions. So if we constrict this one, we dilate this one. If we constrict this one, we dilate that one. Okay, that is known as tubuloglomerular feedback. The tubule is feeding back on the glomerular function. Okay. The other mechanism that will help us protect the glomerulus from undue pressure from the systolic wave is called myogenic autoregulation. Again, the smooth muscle is regulating itself. The muscle is regulating itself. It senses a pressure wave, and instead of relaxing like normal smooth muscle in arteries, it actually contracts. That keeps the flow pressure steady in the glomerulus. And then we use tubuloglomerular feedback for sort of long-term changes in hydrostatic pressure. Myogenic is very, very short-term. It's for those systolic waves. Tubuloglomerular feedback is sort of longer duration changes in GFR. So now we're done with that side of it, and now let's start talking about the other things that go on in the tubule. Namely reabsorption, that is to say if something filters and we want to return it to the blood here at number 2, so number 2 and number 2, reabsorption. We also have secretion. There are things that don't filter in the glomerulus that we want to send to the urine, so we will actively secrete those things. And then finally, the sum of filtration minus reabsorption plus secretion is equal to excretion, that simple formula that I showed you a couple presentations ago. So the mechanisms that are involved certainly involve primary active transport, and that's where the transporter is hydrolyzing ATP directly. We're moving things with active transport either against their concentration gradient. Occasionally we'll move things that just can't get across a membrane at all. Exocytosis or endocytosis type transport, vesicular transport, that would be under active transport. But mostly what we're seeing is primary active transport with carriers. Then we have secondary active transport. That's where we use a gradient that's been established by primary active transport. Like for instance, we use sodium. We use a sodium gradient to move something against its concentration gradient. So that's secondary. And we're even going to see tertiary active transport in the following presentations, probably in 2B most likely. Why do I go all the way to tertiary is because of the folks that are going to get involved in kidney physiology, specifically pre-meds or anybody who's interested in urology. Tertiary becomes a very important mechanism and a major barrier. So I thought I'd give you just a little bit of an introduction. Once you get it, it's not that complicated. It's just a matter of getting the first few steps, and that's our goal with that. So excretion, again, is that sum of these three things. A lot of what we're going to talk about moving forward in these last two videos is circling back to Unit 1. So we have to think about the general properties of diffusion. We have to have a gradient of some kind. We're going to be using kinetic energy if possible. We're going to move things faster if the gradient's higher or the distance is shorter, temperatures and the size of the molecules, and certainly things about Fick's law as well. So Fick's law is very, very applicable in this system because we are doing epithelial transport. So we are going across an epithelial membrane. So if it's lipid soluble, like for instance urea, we don't have any real transporter for urea in the urinary system because urea can just equilibrate on its own into the urinary filtrate. Molecule size depends on whether or not we can transport it with passive diffusion or do we need a carrier of some kind, etc., etc. But it all comes back to that Fick's law yet again. And here's the slide from Unit 1. Remember that the key here is to understand how the inputs, lipid solubility, molecular size, concentration gradient, membrane surface area. We're not going to worry about the composition of the lipid layer per se, but it is diffusion that's applied onto a biologic membrane. So if something's very small and lipophilic, it's going to fly through that membrane. So remember to keep in mind what's going on in the various regions of the nephron. We have filtration going on here, secretion reabsorption here, only resorption on the loop of Henle with water, sodium chloride here, secretion reabsorption. And I'm going to start adding a little bit of detail there, not a ton, but I'm going to add a few more layers there. So just a quick reminder about the volumes that are involved in terms of percentage. If we have 100% entering the afferent arterioles throughout the two kidneys, roughly 20% of that volume will filter and become filtrate after the glomerulus. The summation of resorption and secretion will result in greater than 19% of that material being resorbed, which means that when we excrete, we're going to excrete less than 1% of that which entered the afferent arteriole, and we're going to return into the veins more than 99%. Remember that metanephric kidney was an adaptation to life on land, and so we have to be extremely efficient in that kidney because of the scarcity of water. So let's start working on, again, some regional specificity, and I have mentioned this from time to time. We're going to be doing a lot of resorption in this tubule section. Again, why? Because we want to conserve water most of the time. We are generally losing water to evaporation or the occasional excretion / defecation. We lose a little bit of water there. No secret that the colon is actually a water resorbing organ. In fact, if fecal material remains too long in the colon, it begins to desiccate excessively, leading to things like constipation. In any case, we're resorbing material in all of these things, and two specific things of resorption that I want you to remember are in the descending loop of Henle, we're going to resorb water on the way down because the osmolarity is increasing the further down we go into that loop. Animals with longer loops of Henle have even stronger osmotic gradients the further they go out. On the way up, the filtrate's going to get more dilute. It's going to get more diluted. It's not because we're letting water back in. That's kind of the usual way we dilute things. We usually dilute things by adding water, But in this case, we're going to be diluting the filtrate by removing solute selectively. We're going to be removing sodium, and yes, by extension, chloride comes along for the ride. But sodium is the king in this system. If calcium is king in the heart and chloride is king in the respiratory system / digestive, I won't say that too much, but sodium is the big mover a lot of the time in the kidney. That's why if you have people with kidney problems, oftentimes you're going to see them on sodium-restricted diets. In any case, we're going to be removing that sodium from the filtrate, which makes it more dilute as it goes up. We're not adding water, we're removing sodium. Then we're going to do some other things, and I'll talk about those in a few slides. Since we're talking about resorption and secretion, we are talking about processes of epithelial transport. And we have those two major categories of epithelial transport. We have transcellular, which is moving things through the cytosol. Okay, so across the plasma membrane through the cytosol, another plasma membrane transport, and then we're in a different compartment. And then paracellular, things that must pass between the cells and are regulated oftentimes by the tight junction. The tight junction is passive, but it is also selective. It can be extremely selective to the point where things like water can't get through it. And I'll show you some examples of why I think water, or why I classify water as facilitated diffusion, osmosis, facilitated diffusion. In any case, since we are dealing with resorption here, we are moving from the apical surface, the lumen. Okay, sorry about the goofy writing. It's harder to write with a mouse than it might seem. All right, come on, mouse. The lumen, and we're moving it from the lumen towards the extracellular fluid. It's not the blood plasma yet. It's the interstitial fluid, but it is extracellular fluid, and then we might have a blood vessel here that it would move into after that. But this is resorption, so we're moving fundamentally from the luminal side, from the lumen over here, to the extracellular fluid, and then perhaps into the bloodstream. So let's add just a little bit of regional specificity. And then this figure has a lot of extra information on it that we're not going to need to know all of, but let's just break it down into some simple components. Well, we already know about water being moved out of the loop of Henle that's descending, and sodium that we're moving out of the ascending loop of Henle. So, and what's fun is that in this figure you can see the color. You see that's a light blue, that's a relatively dilute. So when we're in the cortex, it's around 300 milliosmolar, and it gets more and more concentrated as it goes down because we're at almost 2 osmolar down here. Well, as we're going up, it's getting lighter blue again. Why? Because we're moving sodium out of it. With the proximal tubule, you can just say we resorb everything, and that's true. 2/3 of all the volume is resorbed right here in the proximal tubule before we ever get to the loop of Henle. Of the 100% that's resorbed, 2/3 of it happens here, about 1/4 of it here, and the remaining balance through here and here in the collecting duct. So in the distal tubule, we're mostly dealing with pH. You can see that we're moving protons and bicarbonate. Yeah, a little bit of water, and yeah, some other things too. It's, pH is the big one, and water fine tuning. And then in the collecting duct, it's pretty much water. Yeah, we're going to move some other things, but yes, it's primarily an osmotic situation that we're dealing with here. We're absorbing water, maybe to varying degrees absorbing sodium. So everything, water, sodium, pH in water, and water, sort of thing. It's just general ideas on each thing. So these are by far the most specific, if you know what I mean. In resorption, sodium is quite often the primary driver when we talk about moving material, and we're going to move sodium exclusively through a transcellular mechanism involving primary active transport, sodium potassium ATPase. The other ions, it's a mixed bag, or water, it's a mixed bag. So if we have anions, like for instance chloride or other anions that we're resorbing, they'll move either paracellularly or transcellularly. It just depends on the size and which carriers are involved, and that's about as exclusive as I'm going to get with that specific. Water is the same way. We regulate water pretty carefully. It'll move by osmosis, which I call facilitated diffusion, either, again, transcellularly or paracellularly. So the mechanisms of resorption are very much like what we saw with in the lungs. Now this is secretion, and I'm just going to fly through this. Remember that in this mechanism, what we saw was chloride being moved, and then sodium and water followed along for the ride. Imagine, though, at this time that we're moving sodium transcellularly, and then chloride and water are coming along for the ride. So you don't have to reinvent the wheel all that much. You've already done the hard work with chloride secretion in this case. It's a similar mechanism, it's just that it's in a different direction, and we're moving sodium first, and everything else is along for the ride, either electrostatically or osmotically. And to show you how we regulate osmosis as a facilitated diffusion, these different colored dots that you see through here are different flavors of the water channel protein aquaporin. We have actually 8 different flavors, different isoforms of aquaporin. And you can see that we absorb lots of water up here with aquaporins, tons of water on the way down, but look on the ascending loop of Henle. There isn't anything going on here. That's because we don't want water to move at all. If water moved, it would move back into the filtrate. So we don't have any aquaporins in here. That's because we're selectively moving sodium out of this. And then we have some aquaporins down here because, again, we're moving down the medulla of the kidney, which means that there's an increasing osmotic gradient. So it's going to want to pull more and more water out. Just another way of harvesting more water as we send that filtrate down to the ureter. So again, sodium active transport is going to be a combination of primary active transport and facilitated diffusion. Anions are going to follow the electrochemical gradient that we generate with sodium, either a transcellular pathway or a paracellular pathway. Water is going to follow the osmotic gradient if it's allowed to move. So if we have either aquaporins or if the tight junction will allow water to move, it will move osmotically. Other types of molecules will move either by diffusion or by carriers. Again, there's nothing specific here. They're going to follow their diffusion gradients and move to the extracellular fluid when allowed. The next few slides, what we're going to do is we're going to break down that primary active transport mechanism that we do use to move sodium from the filtrate through to the interstitial fluid, the transcellular process. We're going to be using the sodium-potassium ATPase and this epithelial sodium channel called ENaC. The epithelial sodium channel, ENaC, is found only on the apical surface of the tubular membrane cells, and it is facilitated diffusion. It's a channel. It is allowing sodium, which is high in the filtrate. Sodium filters quite readily into the filtrate. It's high in the filtrate, just like extracellular fluid normally is. It's low inside the cell, so this is facilitated diffusion into the cell. Simple enough. And then what we're going to do is we're going to use ATP to pump sodium out of the cell. The sodium-potassium ATPase is selectively located on the basolateral membrane. This is a classic example of a polarized epithelial membrane layer. We've got ENaC on the apical surface and sodium-potassium ATPase on the basolateral surface. And here's where the hydrolysis is occurring. That's primary active transport, pumping in a little bit of potassium that we promptly let leak out. So that's the mechanism. We're pumping that sodium out using sodium-potassium ATPase, and now those sodiums have effectively been moved from the lumen to the interstitial fluid. Now I'm going to show you a mechanism of secondary active transport from the lumen to the interstitial slash extracellular fluid. This is very, very common for things like glucose, amino acids, ions, in particular phosphate is an example of this, and other organic molecules. And certainly glucose is an organic molecule with things like vitamins or peptides, other sorts of small molecules that filter that we want to retain in the bloodstream. So we're moving sodium by primary active transport. So we have sodium being pumped out using the sodium-potassium ATPase. Well, we don't have ENaC here. What we have is another type of transporter. This one is sodium glucose transporter. It's a symporter. Both the glucose and the sodium are moving in the same direction. But you see that we're going high to low with the sodium, so it's going down its concentration gradient, but we're going low to high with the glucose, and that means it's going up its concentration gradient. And if you see that, again, being able to read figures is a big, big skill. If you see anything going up its concentration gradient, that is active transport of some kind. Well, we don't have ATP here, so it's not primary. We know that we're using ATP to pump sodium, so it's once removed, and that makes it secondary active transport. So we're secondary right here. Now, we don't metabolize the glucose in these cells right away, or at least not all of it. So glucose concentration is high here, and then we just let glucose diffuse by facilitated diffusion down this, using this glucose transporter into the relatively low glucose environment of the extracellular fluid. So collectively, this is secondary active transport, specifically here, and then facilitated diffusion here. But collectively, it's secondary active transport and almost identical in all of these different processes. But that sodium, we sustain the low concentration here by investing ATP energy to keep it moving out of the cell. So it's always low. We always have a movement gradient. So my suggestion with that last mechanism that I showed you, or the last two mechanisms, primary active transport and secondary active transport, strongly encourage you to practice drawing those out. Use arrows and draw it on a blank sheet of paper to help you understand and absorb it better. If you try to jump to the end of it without doing the in-between steps, I actually think you're less likely to understand it properly, and you're more likely to forget important steps or things that might change, might throw you off. So work on drawing that stuff out. Other forms of resorption that are transcellular include this passive resorption. This is just pure diffusion. Urea is so small, relatively nonpolar, lipophilic, that it will passively resorb. And in fact, it actually equilibrates between the blood and the tubular filtrate. Plasma proteins, we use endocytosis, so it's a vesicular type transport. There are receptors on the inner surface, on the apical surface of the cells, and if they pick that up, they will form small vesicles that will absorb that material and bring it into the cell, either moving it across the cell or perhaps digesting it or something along those lines. It's endocytosis, transcytosis. So that's it for now. We're at 23 minutes, not too bad. A little bit intense there, though, with those last few mechanisms. Again, understand the regional specificity of what we're moving, and then work on those basic mechanisms, primarily sodium active transport, and then secondary active transport of things like glucose or ions or organic molecules of some kind. All right, I will see you on the flip side in part 2B.